Good morning, student. This is topic number 3, inheritance and variation. In previous videos of this topic, you had learned about the Mendelian inheritance, that is Mendelian experiments, the test cross and back cross, and the deviations occur in the Mendelian ratio. In this part, we will discuss about the chromosomal theory of inheritance. Before starting this point, we will discuss about the first part of this topic in brief, that is Mendelian inheritance. The Gregor John Mendel published his work on inheritance of trait in 1866, but for some reasons it remained unnoticed or unrecognized till 19th century, as communication was not easy in those days. His work was not widely recognized. His approach of using mathematics and statistics to explain biological phenomena was totally new and unacceptable to the new biologist. As continuous variation were observed in nature, the Mendel concept of factors as a stable and discrete unit which control the expression of characters, and that the pair of alleles did not blend with each other was not accepted by the biologist. He also did not know the physical location of factors in the gametic cell. In 19th century, the three scientists Hugo de Vries, Correns and Schemark independently rediscovered the Mendel's work on inheritance of trait. Due to the advancement in the microscopy, the scientists were able to observe cell division and the structure of chromosome under microscope. Then start with the chromosomal theory of inheritance. The Walter Sutton along with the Theodore Boveri in 1903 studied the parallel behavior of Mendel's factor and behavior of chromosome at the time of meiosis. Based on these observations, the chromosome theory of inheritance was put forth by Sutton and Boveri. This theory identifies the chromosome as the carrier of genetic material. This theory states that the chromosomes are present in the pair in the somatic cell. During gamete formation, the homologous chromosome pair segregate and assort independently during meiosis. Thus, each gamete contains only one chromosome from a pair. Nucleus of gamete contain the chromosome which carry all the hereditary traits. Male and female gametes, that is sperms and eggs, carry all the hereditary traits. They are the link between the parent and offspring. The fusion of haploid male gamete and the haploid female gamete restore the diploid number of chromosomes of the species. Chromosomes are important factor in the chromosomal theory of inheritance. The chromosomes are the filamentous bodies present in the nucleus. The term chromosome was coined by W. Waldier in 1888. The chromosomes are visible only during cell division. The chromosomes have ability of self-replication and they play an important role in the heredity, variation, mutation and evolution of species. Chemically, the eukaryotic chromosomes are made up of DNA, histone and non-histone protein. The chromosomes which are morphologically and genetically identical are called homologous chromosome. The first part in the chromosome, that is number of chromosome. The number of chromosome in individual of a particular species are specific and constant. The degree of repetition of primary basic number of chromosome, that is X in a cell, is called as ploidy. The ploidy is classified into two groups, the euploidy and aneuploidy. The euploidy means the chromosome number in a cell is exact multiple of primary basic number, and aneuploidy means the chromosome number is not exact multiple of haploid set. The euploidy includes three groups, the monoploidy, diploidy and polyploidy. Monoploidy means the single set of chromosome is also called as haploid. Diploidy means two sets of chromosomes. Polyploids include triploid means three sets of chromosome, tetraploid four sets of chromosome, and polyploid means five sets of chromosome. Aneuploidy is either addition or the deletion of one or more chromosome in a cell, which is classified into two groups. Hypoploidy means the subtraction of number of chromosome, and hyperploidy means addition of number of chromosome. The hypoploidy divided into two parts, the monosomy and nullisomy. Monosomy means the subtraction of one chromosome from the set, and nullisomy means the subtraction of two chromosomes in the set. This is means there is a presence of zero sets of chromosome. Hyperploidy include trisomy and tetrasomy. Trisomy means addition of one chromosome, and tetrasomy means addition of two chromosomes in a set. Then the next point is the size and shape. The size of chromosome varies from species to species. Each metaphase chromosome varies from 0.1 to 33 mm in length and 0.2 to 2 mm in thickness. Then shape. The shape varies according to the stages of cell division. In interphase, the chromosomes are in the form of chromatin network, and in metaphase, they show the maximum condensation and appear short and thick. Then move towards the structure of chromosome. The chromosomes are best visible in metaphase of cell cycle. During this stage, it becomes highly condensed and short. Each chromosome consists of two chromatids, that is sister chromatids, held together at one point called as centromere or primary constriction. The primary constriction consists of disc-shaped plate called kinetochore. Here the kinetin means thread and core means plate. It is a place where the spindle fibers get attached during cell division. Besides the primary constriction, additional narrow areas called secondary constrictions are present. Some of the secondary constrictions are called nucleolar organizer because they are necessary for formation of nucleolus. The part beyond the nucleolar organizer is short, spherical, called as satellite or sat body. Each chromatid in turn contains a long, unbranched, slender, highly coiled DNA thread called as chromonema. The tip of chromosome is called as telomere, which prevent the ends of chromosome sticking together but attaches to the nuclear envelope. The surface of chromosome shows small swelling called as chromomere. The next point, that is types of chromosome. According to the position of centromere, the chromosomes are classified into four types, that is metacentric, submetacentric, acrocentric and telocentric. The first one is a metacentric. The centromere situated in the middle of the chromosome. Here the two arms are nearly equal and they appear V-shaped during anaphase. The second type is a submetacentric. In this type of chromosome, centromere situated some distance away from the middle of the chromosome. Here the one arm is shorter than the other and they appear L-shaped during anaphase. The third type is acrocentric. Here the centromere situated near the end of the chromosome. One arm is very short and other arm is long and they appear J-shaped during anaphase. The last type is a telocentric. Here the centromere situated at the tip of the chromosome. They show only one arm and appears rod-shaped during anaphase. Then some functions of chromosome. The first one, the chromosomes are responsible for inheritance of characters. They regulate protein synthesis and thus help in cell division and cell growth. They control cell metabolism by directing the synthesis of enzyme. Sex chromosomes are essential for the determination of sex. The chromosomes replicate to produce exact copies of themselves, which are passed on to the daughter cells. These are some functions of chromosome. Thank you.